So the recording.
Yeah, just starting. Just a minute. So once it starts, yeah. Okay. So as I was saying that that yesterday what I wanted to convey through the slides, which I'm just going over right now, is that for this slide that I'm showing you here is primarily to illustrate that you could monitor the change in concentration within a cell by using these fluorescent probes, which are the ones like the MAG Fura 2, and you could uh, by by looking at how these stores work, could image the amount of calcium that is there, and you could see the puncta which is there showing you the calcium stores, and how through the coloration that occurs when you by looking at the change that the fluorescence that takes place, you can essentially look at how much calcium is being released. And what I said was that the amount of calcium that is there uh, and the way it is released is uh, very crucial, because it essentially controls a lot of functions within the cell. And then you have to keep in mind that there are three main components that are there: the leaky channels which are there, the that brings in calcium from the environment into the cell and also releases some amount of calcium which is within the cell to the outside. Plus, you have calcium which is there in the cytosol at any given point of time when an event is taking place. And besides that, you also have calcium in the various stores, primarily in the endoplasmic reticulum or the sacroplasmic reticulum or the smooth reticulum, right? So, so these studies are important because it allows you to understand how this calcium functions and what it does. But keep in mind that the concentration, which is the basal concentration, is very low, because calcium is a secondary messenger, and when it is released within the cell, it actually will activate certain kinds of functions within the cell, which range in the nature and functions that the cell can actually activate. So, given that in mind, what we had what we wanted to see was how does this actually happen, and you have to be able to appreciate this fact that because from just taking off from what neurons essentially how neurons essentially function, that you'll actually have both temporal as well as spatial aspects to keep in mind. And there are these release events which take place, which are based on triggers from the outside, as well as it could also be because of signals that are electrical in nature, in neuronal cells, for example. And also there are secondary messengers and the localization of secondary messengers which are important. And so you have to release calcium in a very particular fashion so that you actually get the action that is required within the cell. Now, so that in mind, now the question that people have been trying to understand, and this is where I was fumbling a bit yesterday, is to understand how the calcium is actually getting released inside the cell. So the contention that people try to explain through this particular uh, paper is that well, is it actually all or none release, or is there a graded response, or if it is graded response, what kind of response is it? And so so the proposition that is being made, and which is actually more or less validated, is that actually it is going to be dependent upon a feedback mechanism, a positive feedback mechanism, based on the receptors that are there on the surface of the sacroplasmic reticulum or the smooth reticulum, right? And these are receptors that are actually sensitive to inositol 3 phosphate. And the inositol 3 phosphate receptor, which is the IP3R receptor, which is there on the surface, which essentially activate the amount of calcium that is getting released into the environment. So that is one kind of understanding that is there. So the first layer of experiment that was done here is actually, if you look at this one, they did it in a single muscle cell or myocyte, and that's the image of that. And the image is taken over different conditions, and the conditions that are there are given in A and B. One is that you have a calcium-free bath solution, and the other one has calcium in the bath solution. And of course, the carbachol, which is here, is essentially an agonist, which is similar to the muscarinic, receptor here. In this case, that is how the calcium is going to be released when this will go and attach to it. So there are two different concentrations that are taken. One is at 50 micromoles and the other one at 500 micromoles. And what they're showing you is that once you release it, it goes down to the basal level after some time in a calcium-free bath. And then if you increase it to a 500 micromolar, then it again increases. And the reason why that happens is because not all the calcium that are there in the stores is released at one point suddenly. So it depends. So there is a response which depends upon the amount of the carbachol which is present in the experiment, or you actually dose in the experiment, and you see that it goes back gradually to the basal level, and of course this reduction is a little slower after a certain level, and because there is this initial increase that was there, so the sustained amount, although it is continuing here at 500 micromolar, it does not sustain the signal. It has a spike, and then it releases to a small amount, which shows that not all the control is with the sacroplasm, within the stores being released all at once, because that is, if that was there, then everything would have been released, and then you would have nothing left. But that you had two different peaks here, as well as in spite of all the signal being present on the outside, there is a control because it does not release all the quantity of even at this level, it does not release all the calcium that is present in the cell. Now, if calcium is present on the outside, then you start seeing this kind of oscillation. And this is primarily because you are also having calcium from the outside coming into the system. So within the cytoplasm, when it enters, when you're measuring the current or you're looking at the fluorescence of this, in this case, then you start seeing these oscillations. Now, this kind of oscillation is something that is important because that is also a mechanism by which this calcium is released, because it essentially will release as either like a spike, depends on that is really at very low concentration. At intermediate concentration, it will be like a puff. That means it will release as a big amount, but then it will stop after some time. And then you have these waves. So it keeps on happening one after the other. And you get this kind of oscillatory in nature behavior which is taking place.
So
and that is what was explained in this behavior.
So what would be the effect of those like oscillatory response of calcium concentrations?
Yeah. So there are certain, so calcium, when calcium is released, it essentially affects many different kinds of, systems, rather receptors and other proteins which are there, which respond to the calcium signal, right? And the signal response is of different kinds. It responds there are, for example, calcineurin will be there. It responds both to the level of the calcium that is there, the concentration that it reaches, that wherever the calcineurin is there within the cell, and also to the frequency at which it comes. So, and based on that, there are certain cell cycle responses or gene regulation responses which are there, which can only happen if that kind of a certain kind of frequency is there. So it is tuned to that, and that's quite advanced kind of coding for how these proteins behave. And then you'll have durations. So something that is there will be what is the level and what is the duration. So some proteins will not respond if there is just a certain high level of calcium for a short duration, but it might respond to a lower level than the lower level, but which is sustained, and the opposite. So these kind of behaviors are there within the cell, and that is the really reason why this kind of oscillatory behavior is something that is observed. And for example, in astrocytes, these kind of when there's an associated neuronal activity in a certain region of the brain, when you start seeing high activity, then along with the action potentials that are being generated by these nerves which are firing, you also release calcium which go in along with the same frequency or nearly the same frequency, and or something that that is coded with that frequency, and so you start seeing these waves. So that means if you're at one point in a certain region of that brain, then you'll find that you're seeing these frequencies. It'll be like highs and lows that you start seeing at that point. But if you looked at the overall spatial distribution, then it'll be like waves going, right? But if at one point, then the wave keeps hitting you, and then you see the spike going up and down, right? So that's the way that the behavior takes place. And because of that, it is able to control the capillaries that are there in that region of the brain, and they can expand the capillaries so that more blood flows into that area. And because more blood flows, you have more oxygen supplied to that area so that the neuronal activity continues, because that is what is being demanded. And that is again picked up by the fMRIs, which is the bold signal, blood oxygen level difference. So you have a basal level, and then the sudden change. Now, how much has it changed by? You can do this color coding, which is done by software, and then you can see the reds would be a high region and the blue would be a low region, and that's how you map the whole brain. For example, they do different kinds of tests, cognitive tests when people speak. And or they are looking at a screen and then identifying and then clicking on doing some kind of cognitive jobs, then certain parts of the brain will start lighting up if they are under the fMRI, right? So or even that.
So are these
yeah.
Are these oscillations like uniform oscillations, or are these like random spikes and drops?
Random? No. So when the oscillations begin, so when see the frequency of coding is dependent upon how much of the ligand is there in the environment trying to do this. So depending upon the circumstance, the frequency of this will change. So you can have a spike which is at very low. That will be just a single peak response, depending, maybe a high spike or maybe a small spike. But when you have a sustained agonist or a receptor or the ligand in the vicinity of the receptor, then they keep on sustaining that event that is taking place. That means the release of calcium. Then the dynamics of that is such that it starts giving you this oscillatory behavior. They are not random, but they will differ depending upon the cell type, rather the receptor type, also the cell type actually, and where it is and how much of that is being present. So it is not a random thing. So the waves that you see would have a certain frequency, and that frequency is important also.
Sir?
Yeah.
So is the frequency of the oscillation depends on the intensity of the signal?
No, intensity of
the signal.
Yeah. The frequency. The frequency will depend on the intensity is not the magnitude. So, what do you mean exactly? So let me ask you, what do you mean by exactly by intensity of the signal? In this case, what are you looking at? Are you looking at the ligands, or are you looking at the signal that the ligand is generating?
So the number of ligands, the ligands are.
Yeah. So if there is an excess of ligand in the environment, of course there is a certain frequency at which it will saturate, right? There will be because there is a certain balance about how much the cells, the stores can actually release at a given point of time, how much is getting removed by the leaky cells, and plus how much is there in the cytoplasm doing the action, right? And so that all of that together will actually have a balance. And so although it depends primarily on the signal that's coming in from the ligand, but there are other factors that will control the dynamics of this. So it's not just a one point or a single thing that is actually controlling the frequency of oscillation.
Okay, sir.
Okay.
So one more thing. So how would you differentiate these like oscillating signals from something like seizures? Because I guess even in seizures, the action potentials like randomly spike.
Yeah. So that when you have seizures in the brain, actually usually what happens is you have a situation that there is a malfunction in some of these receptors of calcium, if it is due to this, if it could be for other reasons. Let's say if it is due to some kind of a calcium signaling problem, then essentially it means that there is some kind of a malfunction that takes place, right? So it could be because of so many things, So it could be because locally maybe the myelinated sheaths are not there, something has happened, or some other that maybe because of some kind of a genetic mutation, or a proclivity of this patient to have seizures, who is having the seizures, there is an excess of activity because of some kind of a trigger that happens from the environment, right? Could be a high, it could be a smell. So some kind of an aromatic compound which is there could actually trigger sometimes. So people get migraines from these kind of events also. So migraine is also some part, like, in a way, something that is also an event that is linked to these kind of things. So whenever you have a seizure, what happens is you kind of have an overactivation of these of the calcium release that's taking place, and so what is happening is essentially because of this uncontrolled release that is taking place, there is no control, where essentially you are stopping the activity that is being initiated by the calcium ions that are getting released in the vicinity of that location from where the calcium ions are getting released. So obviously, the calcium ions, when they release, they have certain functions, and if for example, they are activating astrocytes, they will continuously be opening and closing, locally, expanding and contracting maybe the capillaries which are there, and obviously, that could create a problem. So the seizures are usually not that. I mean, that's one of the ways in which these things can be explained. There are other explanations, but if that answers, then we can. Is it sufficient?
Yes, sir.
So what this particular slide shows is, essentially that inositol 3-phosphate could actually bind to these receptors and release calcium from the lumen into the cytosol. So what they are trying to say is that, well, in the presence of this IP3, you will perhaps, for example, activate in this case, in this locality, one of these. The way to illustrate this is let's say you take one of these receptors which are getting activated, and so there's a depletion of calcium in this bluish zone to release of calcium in the reddish zone. Now, even and if you continue this, although this the region expands, it will not be able to release as much calcium, because obviously, there is a control which is there by the cytoplasm. So there are, but if you were to increase the amount of inositol in the vicinity, then this calcium that is getting released is actually going to influence the receptors in the vicinity, and is going to enable another such receptor to also release calcium. And this is called the calcium-induced calcium release that takes place, in which, although this cytoplasm, which is reticulum, or the smooth reticulum which is here, is actually going to be releasing calcium into the environment, there is this control which is taking. There's going to be a mechanism by which this is going to get controlled, because you can't have complete depletion of this. So although you are having excess of this, there is, and this would induce a release. The moment you find some decrease in this IP3, immediately this will stop the release of this calcium which is in the environment, or there is a certain amount that can be depleted, and even in spite of that, you can't really keep on releasing the calcium. So at some point, you're going to kind of saturate that signal of calcium that goes through. And this phenomena which is there, if this is really captured properly, then it can actually, if you model that, then you are able to describe some of the phenomena which is observed for calcium release. That is, when there is a sustained release, you could get a huge amount of calcium which is there in the cytoplasm, or if you're looking at neural cells, then into the extracellular space. So those kind of studies have been done, and there are these paradigms which are there also. So you can have a neurotransmitter, which neurotransmitter secretion. So this can actually lead to neurotransmitter secretion. It can lead to contraction of striation muscles. It can lead to smooth muscle contraction, like the heart muscles and all that. And then it can also lead to the adrenergic control of the heart contraction. So there are many other details which are there. But here, if you were to just look at KEGG pathways, and if you look at the KEGG pathways and go there and try to understand this calcium signaling, then what you'll find is that that one of the regions where you would find the signaling becoming important is that, of course, associated with the receptor, as well as there's a protein kinase which is there, protein kinase C, and calmodulin, which is involved in all of this. So this is usually the most fundamental aspect of this calcium signaling, but there are others. So if you were to click on this region of KEGG, then it gives you the next slide that I'm showing you here, and this is the detailed calcium signaling pathway, where it shows you what is bound on the receptors that could participate in this kind of activity, and then how it actually goes forward to give you a release of calcium, which then affects a series of these other downstream proteins to give you various kinds of actions, and including apoptosis. All right, and if you look at apoptosis in detail, then you'll find that you have a IL-3 which will bind to the IL-3 receptor, and if you move down that, then you'll find that it gives you a survival signal. Whereas if you had the excess of calcium that was already there, and that leads to some stress, then that gives you the apoptosis here. So there are two balances which take place. So obviously there is this always balance between survival and apoptosis, which is continuously being monitored, or rather it continuously happens inside the cell, because and of course it's quite complex, and not all of this can be explained just by simple modeling, because the model has to capture the behavior as you observe in experiments, based on what is there in individual components of the model itself. That means you would have to build up and then see if there is an emergent property, such as, let's say, the oscillation of a calcium wave. Can you do that? So that has been a major cause for study and modeling for calcium signaling. So let's look at one other aspect here. So, and this is sometimes this failure analysis kind of analysis, which is done for various kinds of networks. If that is done, then you seem to get some more understanding of what really happens. So you could have a material flow failure. That means you could have elevated blood glucose, or you could have elevated blood glutamine. So and this response, how does it behave? You could check that out, and also you could look at, let's say, signaling failure. That means once the signal has come into place, is there changes in binding affinity? Is that how is it? Is it robust? Is it safe? And then changes in the calcium to calcium signature. Is it sensitive? Can it cause a disorder? So those are questions you can ask once you've done the model. Questions so far?
None currently.
Okay. All right. So this is a model that I had constructed a while ago, using some basic models that are there in the literature, and then using that to construct a Simulink model. And what I was trying to see is that if you had a sustained calcium signal, what would the effect of that, right? So and if you see here, if you just looked at the oscillation of the signal based on just adding some ligands and then looking at it, and you were looking at, let's say, a particular output from one of the proteins there. Okay, so and you were looking at, let's say, the oscillation that are taking place in the cytosol, which is the CaC2. So you can see that there's an oscillation there, and then if you were to not give a very high concentration of and in the absence of this signal, which is the BAD signal, okay, Becklin associated signal, which is there, if that is not there present and it comes in later, you'll find that it doesn't really affect this oscillation. So the oscillation is there, and after some time this oscillation dies out, and this has no effect on that. But if you had ahead of time, if you had given a very high, if you had a very high concentration of calcium in the cytoplasm, then it shows you that the error levels are the same here. So what I've not shown here is
could
actually.
You explain these graphs once again?
So what you see here is the calcium signal that you would observe if if you had the failure of, let's say, CICR. So what you do is essentially you go back, and one of the proteins which are participating in this, if you kind of mutate that digitally and let this simulation take place, you can check out if there is a change in the behavior of the oscillation. So when the model is all perfect, then what you'll see is that you get an oscillation which is typical of a calcium signaling oscillation. So there is a very periodic oscillation that you would observe. That is an expansion of this, okay, this region. And if you expand that, you can see this. And in this case, what happens is you don't see any signal of this BAD coming up, for a long time. And so obviously, you assume that what we are saying is that no, we are looking at simulation conditions, and we are saying that, well, it takes a very long time for you to have this signal on to allow this signal to go up, which is, in this unit, it's like 3,000 seconds before you can see this trigger going up. Okay, so this triggers up according to the model. Now, if you did not have that, so you can see the level here is about 0.4. Whereas, if there was one of these proteins were not functioning, either calcineurin or calmodulin or some of them, so if they're not working properly, it's not working properly, then right from the beginning you'll have a high amount of calcium in the system. So you don't see the oscillation structure anyway, first of all, and then you start seeing this high amount of calcium that is coming up, which is what I said, that you have a sustained amount of calcium in the cell in this case. And because of that, you'll find that, well, this bad signal which is there, it triggers in much earlier. Okay, and once this triggers in, this is going to essentially, so there is a certain amount of delay, and this delay is essentially there in the coding of the, the coding of the cell itself. Because the reason is because when you are looking at the parameters that are used for this kind of a model, you're taking the parameters from a certain organism or a certain cell type, and those are from experiments, and you're using it. So by evolution, you'll find that these parameters are such that if you allow them to evolve in a simulation time, then they will all behave as if in that simulation time, as if something similar would something similar would have happened inside a cell. Now, if I'm looking at a situation like that, and I'm of course resetting the zero time to the time where where the initial or the first time I see the calcium signaling going up. Just like here, when the initial time is set to the first time I start seeing the oscillation, that is the initial time is also taken here. So the first time that you see the calcium signaling, the oscillation is missing and it goes up. So if one of those proteins are shown to malfunction, that means you either set it to zero or you change one of the parameters to some degree, and you can play with that, or you remove that from the signaling pathway, then what happens is you'll find that the signal that you observe is actually a sustained signal. And this delay that is there, this delay is actually something that is part of the coding of the response. And you'll see more of this in this course, where these wiring diagrams essentially are coding both the spatial and temporal events. So here the temporal event is that there is a delay. So in this period, if this changes, let's say by some repair mechanism, then this will not happen. And again, of course, here also we are doing it artificially, we are sustaining it. Here also, we would assume that in the cell, before this event triggers, some other event will take place, which will essentially reduce this calcium to a level, which is the basal level that is acceptable in cells. Okay, is that clear now, Nishantan?
Yes, sir.
Hello. Okay. So there are other aspects of calcium, and one of the places where it is seen is in the work that I do in my lab is in ALS. I'm not going into details of this. I'm just going to show you that there's a hub protein which is here, which is the androgen receptor. And if I go down to this, I'll see that right here. So PSEN is another receptor which is important, and this is part of the calcium signaling pathway. So if this is malfunctioning also, you'll start seeing problems with the, I'll show you another simulation with the PSEN. But this receptor is actually, we had a lot of interest in this one protein called angiogenin, which essentially it codes for blood vessel regeneration, and along with other, it's not by itself, but along with other genes. But it's very important because if this is not functioning, then you tend to get ALS. Of course, this is just one of the many things that can happen, and there are more than 50 genes that are implicated in the disease now, and more than 150 risk factors and other things which are there. So we're not looking at all of those details, but this PSEN that is there also plays a role in calcium signaling. So I'm going to skip this part, go down to this. So if you look at the way this works out, oops, sorry. I want to move this out a bit. So this actually has two-pronged effect. One is the calcium signaling and giving you cellular signaling. The other one is it affects gamma secretase, and that affects the NOS signaling, and then the APP processing. So so this is the location of this. It's essentially in the reticulum here, and that's the endoplasmic reticulum, I think. And so if you were to model how this affects the calcium signaling, so along with this, so this entire pathway was modeled, and all the details are given here. And along with this, we also looked at leaky calcium signaling and release of calcium, from this and the cytosolic calcium. So, and if you just look at the phase diagram, like I was telling you earlier, so you could look at calcium from the ER, endoplasmic reticulum, and the calcium in the cytoplasm. That's one kind of look you can do, and the other one is in the mitochondria versus the endoplasmic reticulum. Okay, so those are the two phase diagrams. But you look at the oscillatory patterns here. So the oscillatory patterns are very regular when things are all fine. So and the phase diagram will also show you this kind of behavior. And of course, what you're looking at is changing levels of one of the parameters I changed. I forget now which parameter it was. It's been a while. So, but what you notice is that if the concentration of this of the calcium is monitored, then this regularity starts getting degenerated over a period of time. So one of the parameters that is shown here is one of the parameters that affects the function of PSEN. And so as you change the parameter, you start seeing a behavior which is essentially degrading, and you're losing much of the characteristic that you would see otherwise. And so that shows you that so this kind of a cycle is showing you that the cycle is stable, and of course that is also observed in the oscillating nature that you see here. So this is a kind of behavior that you can also go back and use some kind of control theory to explain, but I'm not going into the details here. So with that, I'll stop and ask the class to tell me if there are any questions that you need to ask at this point. You can come online and ask me.
No specific question currently.
Okay.
So, but I found like this paper which actually details the role of like I was asking you yesterday, what role does carbichol play in the calcium
yes
level? So I found this paper which actually details it quite well.
So is the same paper, the citation, the same paper I gave, or a different one?
So I think it's a different one.
Okay. Okay. Who are the authors?
It's in the Journal of Physiology.
Okay. Can you then share it with the group here?
Yes,
post it.
Put it on the chat.
Yeah, post it on the chat, and so that people can also look at it, and I'll also take a look at it. The intention was not to go into details of that, but it's a good thing you asked, and that always goes to show what I put as a disclaimer in my first lecture, that I can't remember some details sometimes. So I'll just until I'm very regular with it or doing work with it, I will not remember certain details. So, but good that you asked, because it. And yesterday, actually, let me turn off the recording, then I'll just talk about it a little bit. Okay, give me a second.